Our project is actually entitled Characterizing and Determining the Anti-Cancer Properties of Selected Vegetables and Mushrooms in the Philippines. And this is actually an interdisciplinary project which involves the faculty from the chemistry department and biology department. And it all started when I realized that the chemistry actually have so many extracts from plants and vegetables, and they just store it in the refrigerator after they have extracted and characterizing all of these plants. It just stopped there. So I was thinking that if we can form a group that would look into the chemical properties as well as the biological properties of these plants, it would be a good idea. Those vegetables actually are very common ingredients of the everyday food that we eat. But more especially, this group of vegetables belong to what we call the cruciferous vegetables. This group of vegetables have special secondary metabolites we call glucosinolates. And these glucosinolates, when we eat the food, we chew the food, actually broken down to chemicals we call isothiocyanates, which are believed to have many medicinal properties. And one of the medicinal properties we would like to study is the anti-cancer property of these cruciferous vegetables. The project actually does not aim to identify a potential drug compound, not yet. No. We just want to have we always hear these anecdotal stories about this food is good for stomach ache, this food is good for toothache. We wanted to have an established scientific basis of us claiming that these particular extract have medicinal properties. In scientific studies, before you do that, you should do cytotoxicity studies. Cytotoxicity meaning to say, when you add it into cells in culture, they are harmful, they will cause the cell to die. And then the genotoxic would look into a higher level of genetic consequences or genetic effects. Genetic would mean, I don't know if you've heard about DNA, the genetic material in the cell. So cytotoxicity, we use cell lines. We use cell lines because we cannot test this yet into humans. So you do it in cell lines so that the ethical issues are not there. These are cancer cell lines which have been grown from tissues of cancer people, cancer patients, and have been purified in the lab. We have found that several extracts, their cytotoxicity level would differ from one cell line to another. The group also looked into the effect of the concentration of that secondary metabolites in different food preparations. For example, the labanos. If you make salad of labanos, you make labanos in the sinigang. And they found out that the concentration of those metabolites actually changes as you do different food preparations. One of the goals of the project is actually to come up with a monograph of these vegetables, complete with the pictures of the vegetable, an instruction on how to prepare the vegetables, the manner of cooking that it can be done, so that people would know what kind of food preparation and what plants or vegetables they will have to eat, so that they get the maximum benefit of the compounds that are found in the vegetables.